Hi, this is the Combined Science Trilogy Foundation Tier Biology Paper 1 for the 2024 season. Right, this is then question number 1. Right, so this question then is about cells, and what it does, it shows an animal cell. Okay, not the typical one that you might see. And what it's doing is asking for A, B, C, label it. Now, the most obvious one to me then is, and this at the top side here, what we've then got is we've then got all the words that you can possibly use. So B is obvious. Okay, so what I'm going to do is I'm going to put nucleus. Okay, A is then the area around the outside, and what they've tried to do is they've tried to catch you out a little bit, and they've put cell membrane and cell wall. Right, so don't forget cell membrane, right, is in an animal cell. So cell membrane. And C, which is kind of like the jelly-like substance, is then the cytoplasm. Okay, what is the function of the nucleus in a cell? Okay, we've just done that, so we've just got a nucleus in there. Contains a solution called cell sap. Now we know it's not that, that's a plant cell, right, and it's the vacuole, so it's not that one. Control the activities of the whole cell, yes. Okay, control the movement of the substance into the cell, that's the cell membrane. What is the function of mitochondria? Okay, so what we've got is we've got an animal cell, we've got mitochondria in there, we've got ribosomes in there, we've got the cell membrane around the outside, we've got cytoplasm, mitochondria. To produce glucose using photosynthesis? No, because photosynthesis is purely in plants. To produce proteins, proteins is ribosomes, right? Protein synthesis. To release energy from respiration, yes. Okay, right, that's a little bit smaller, right? So 1.4 shows a light microscope, okay, standard picture that we've seen loads and loads of times before. A student is given a prepared slide of an animal cell to view using the microscope, okay, prepared slide, okay, so that means it's already ready, okay, of an animal cell. Complete these sentences using labels from figure 2. Place the slide on the, okay, well, it's got to be the stage. Okay, that's where you're actually placing it. Use the objective lens that has the low power. Okay, so these are then the objective lenses there. Shortest one is the lowest power. Use the objective lens that has low power. Look through the eyepiece lens. Okay, make sure you're writing all of it down, even though my handwriting is shocking. Direct the light by moving the mirror. Okay, often what you'll do is you'll have a light bulb, okay, but this is then a mirror. Make the image larger by losing the high power objective lens. Okay, so the longer the lens, the objective lens, the more power, right, the more magnification there is. Right, figure 1.3. Figure shows a different animal cell. Okay, calculate the magnification of the image in figure 3. Calculate the image, right? So what I'm going to do is immediately they're talking about the AIM triangle. Okay, complete the following steps, 4 marks. Right, measure the width of the image in millimeters. Right, so then what you got to do is, so I'm just looking around for a ruler now. Right, so then what you do is you literally get a good old ruler. Okay, and then what you do is you do Y to Z, right, and you do it in millimeters. Now I've done that, and that says 36 millimeters. Give the width of the image in micrometers. Okay, so therefore, if 1 millimeter equals 1,000 micrometers, we're going to do 36 millimeters multiplied by 1,000 gives us 36,000 micrometers. Now make sure you write it on the line. Okay. Figure 3, so the one we were just looking at, has a real width. So I'm going to write my triangle here again. Okay, so it's AIM. Actual image magnification. Use the equivalent, they've given it anyway, right? I like this one though because it's kind of what I always think about all the time, right? The cell in figure 3 has a real width of 40 micrometers, okay? So therefore, the real width is this bit at the bottom here. So calculate the magnification. So magnification, I always write it out in full, right? It just kind of, plays with my head that if I don't write it out in full. Width of the image was 36,000, we did that before, divided by the real one is 40 equals 900. So therefore magnification is 900. There are no units with it, right? And it actually says on the specification, the answer scheme, if you write units, you don't get any marks for it. Right, question number 2. A student investigated the loss of mass from leaves that were placed in winds of different speeds. Okay, I assume the wind's coming from this. The student used an electric fan to create different wind speeds. This is the method. Record the mass of one leaf taken from a plant. Okay, well, that's interesting. One leaf. Attach the leaf to a stand. There it is attached. Leave for 1 hour with the fan off. Okay, that's to get it acclimatized to whatever it needs to do. Record the final mass of the leaf. Right, repeat the steps 1 to 4 with the fan set at different speeds. Use leaves of similar size each time. Okay, so that's going to be tricky, getting the same leaf each time. Calculate the loss of mass of each leaf. Okay, right. Okay, so table 1 shows the results. Off, that's the loss of mass. Low speed, medium speed, high speed. Okay, why did the student do experiment with the fan off? Right, so that is then as a control. Okay, what you've got to be able to do is you've got to be able to compare to the other results. Okay, now what I've done then is I could have said as a control and they're going to accept that. Right, but what you've also got to do is you've also got to make sure that you clarify it. I'll always say every single time, even if it's a 1 mark question, don't just write the word control, even though you're kind of right. Make sure that you justify what you're actually saying. Okay, so I'll always say no answer ever only has 1 word in it. 2.2. How does increasing the fan speed affect the loss of mass from the leaf? Use table 1. Okay, so what we've got is loss of mass increases as wind speed increases. Oh, I've forgotten how to write. Okay, so what you could do is you could just say loss of mass increases, okay, but it means nothing. Justify it, clarify exactly what you're talking about. 2.3, okay, explain. Oh, a good old explain question. Okay, 3 marks. Why the mass of the leaves decreased at all fan speeds. Okay, so what it has is what you're going to do, firstly, you're just going to describe what you're actually talking about. So the loss of water has mass. Okay, so when the water leaves the leaf, what it's got mass. Makes sense. Okay, then you're going to go so, right? So if you've got an explain question, you always want to kind of justify something with so. So water has evaporated. Well, evaporated from the leaf, and you could also have transpired. Okay, and I'm clarifying it again, from the leaf. Okay, you could also have water has left from the stomata. Okay, so key thing here, explain, do a little description, and then you go so. So, it's dead important. 2.4, right? The student repeated the investigation in a room at a higher temperature. Complete the sentence, choose the answer from the box. Okay, so we've got 3 answers. At a higher temperature, the loss of mass from the leaf would be, well, greater. Okay, because there's more evaporation, there's more transpiration going on. 2.5. How could the student check the repeatability of the results? Okay, so repeatability means, is it going to be the same every single time? Do the investigation, oh look, 1 box. If you tick 2 boxes, say goodbye to the mark. Do the investigation again using a different method. Stupid. What's the point in doing it with a different method? Do the investigation again using a different type of plant. What's the point in that? It's not comparable. Do the investigation again using the same method. Yes, because then what you do is you're repeating it. Okay, that's what repeat means. Right, figure 5 shows different parts of blood. Different parts of blood. Okay, it's quite weird really, isn't it, looking at those? All right, so we've got just the liquidy stuff. then we've got the smaller cells, then D is then tiny little cells, and then C, it looks like they've got a nucleus in the middle or something. Which letter shows a white blood cell? Right, well, platelets are fragments of cells. Red blood cells don't have a nucleus, so therefore the big ones, which letter shows a white blood cell? It is definitely C, right, because C is a white blood cell and it has a nucleus. So A is going to be the plasma, it's like the yellowy liquid. B is a red blood cell, and D is a platelet for clotting. Which part of blood causes the blood to clot? Okay, I've just answered that. It's not plasma, that's the yellowy liquid that carries all the hormones and nutrients and urea and amino acids. Red blood cells carry oxygen, so it is platelets. 3.3, right? Name 2 substances transported in the blood. Okay, I've just mentioned them again. Okay, we've got oxygen. Okay, now that is oxygen in red blood cells. Okay, loads of other ones could be hormones. Urea is your waste product, which gives it the yellow color. Carbon dioxide is transported in the actual plasma itself. Water, glucose, amino acids. Okay, there's loads and loads of different things that could be transported. Healthy people can volunteer to donate blood. Okay, last time I donated blood 10 years ago, I fainted, which is a bit stupid. Okay, quite embarrassing as well. Healthy people can volunteer to donate blood. The donated blood is then given to patients who need the blood. Okay, what would be the point in donating if you didn't? When a volunteer donates blood, the volunteer's health is checked. Okay, you don't want anybody ill. A needle is used to remove the blood from the vein in the arm. Okay, the volunteer rests for 15 minutes after the donation. Okay, so I rested 15 minutes, had a cup of tea, and then fell on the floor. Suggest one reason, one is in bold, why blood is removed from a vein and not an artery. Okay, so a vein is low pressure. Okay, so a vein is low pressure, all right, because if you then put a needle in an artery and it's under high pressure, then you might have a bit of an issue. Veins are also easier to puncture. Okay, so if you think about the actual size of them, so there's an artery. Artery's got a big, thick wall. Veins have got a much bigger lumen and a thinner wall. Veins are also easier to access, right? They tend to be closer to the surface. And because of the pressure, right, veins are also easier to stop bleeding. Okay, because again, if it's under high pressure, right, it's going to come out faster, so then you've got a bit of an issue. 3.4, right, oh, I've just done that one. Right, suggest one reason why blood is removed from a vein, not an artery. Okay, patients may receive blood during a medical emergency. Donating blood and receiving blood has risks. Okay, so we don't need to answer that one again. All right. Suggest one advantage and one disadvantage to a patient who receives blood. Right, so an advantage. What you're going to do is you're going to improve the quality of the person's life. Quality of life. Okay, an advantage is if you give blood, you're going to save lives. Okay, and also the advantages are you're going to restore blood levels for the person who's getting the blood. Okay, makes sense, doesn't it? It's great. Disadvantages, okay, so let's do it in red. Let's be mean, okay, is a risk of infection. Okay, so if you then get blood taken out of you, there's obviously a puncture wound in your arm, you could get an infection. You could get an allergic response. And you could get bruising. Okay, so even when I gave that blood that time and I fainted, I had a huge, great big bruise on my arm. Right, 3.6. Okay, suggest one advantage and one disadvantage to the volunteer who donates the blood. Okay, because the one above here was who receives the blood. Okay, donates the blood. What you are doing is helping people. okay, which is fantastic. And then you are saving lives. Okay, a disadvantage is it can hurt, pain when donating. And also you've got risk of infection. Because again, if you've got a puncture wound, just like previously, if you've got a puncture wound, right? And then again, it's the same sort of thing, bruising where needle went in. I can't fit it all in, so needle went in. All right, so remember the difference there. You've got donating, okay, donating and receiving. So make sure your answers are thought through. Question number 4. The question is about a disease, gonorrhea. At least I've got to ask you how to spell it. Right, gonorrhea is an STD, sexually transmitted disease. Which is a symptom of gonorrhea? Okay. Damaged immune system. No, I think they're talking about AIDS. Okay, HIV. Pain when urinating. Yeah. Reduced body temperature. No, I don't think so. It's not like a main thing of it. So pain when urinating. Gonorrhea. Ooh, table 2 shows the number of reported cases of gonorrhea in 5 cities. Gosh. Number of reported cases of gonorrhea per 100,000 people. Okay, so if you've got 100,000 people in City A, there's 124 cases. The number of cases of gonorrhea in each city might not be accurate. Okay, so it might not be accurate, right? Let's do some purple. Not all cases are reported. Okay, so somebody might get treated for gonorrhea and they might not get reported. So therefore the numbers are going to be lower. Populations of cities are estimates. Okay, you don't exactly know how many people are living in Manchester or Preston or wherever it may well be. You don't really know. 4.3. Which is the best way to present the data from table 2? Okay, it's not really a line graph, is it? Because they're all different size cities. So the best way of doing it, bar chart. 4.4. The data is given as per 100,000 of the population and not as the total number of people with gonorrhea. Okay. So the whole idea of this is compare cities. Okay, you couldn't do London, could you? London has got a population of however many million, all right, and then compare it to, I don't know, Blackburn or something like that. This might only have 100,000 people. I don't know how many people are in the Blackburn. So the numbers are obviously going to be less. So you have to do a comparison, which is per 100,000 people. So it allows you to compare cities because different cities have different populations. Okay, or else it'll always make the biggest cities look way worse than everywhere else. Oh, coronary artery. Okay, coronary artery. This is something that always gets people confused. Is right. So this is a typical heart, obviously. Okay, a little romantic heart. A coronary artery is actually one of the arteries that's on the outside of the heart. Okay, it's not anywhere else, it's on the outside. Okay, and they are the arteries then that are then delivering the blood to the heart muscle itself. Right, deposits can form in coronary arteries. The deposits are called plaque. Plaques cause damage to the arteries. Figure 6 shows plaques in a coronary artery. There we go, a bit of plaque there. Sounds a bit like your teeth, doesn't it? Plaque. Okay, and there's a coronary artery. Which 2 factors increase the risk of plaque forming in the coronary artery? High fat, without any shadow of a doubt. Blood glucose low? No point. Regular exercise? No. Smoking? Yes. Taking statins, that reduces the amount of plaque. So high fat diet and smoking. 4.6. Which treatment can be used for plaque? A placebo, that is a drug with, or a it's a medicine that's given with no drug in it. So how can that possibly work? A stent, that's brilliant, right? Stent, so there's your artery. And what happens is they actually put a balloon in there first with a little bit of chicken wire. There's the chicken wire, right? The balloon opens up the artery and then leaves the chicken wire in. And that's the stent. It's kind of like a very, very thin bit of wire. So it's definitely that. Antibiotic is for bacteria. 4.7. A heart attack can happen when plaque blocks the coronary artery. So there's your artery, there's your artery, there's your plaque, and eventually what happens is this area here will get blocked. During a heart attack, muscle cells in the heart die. They're not getting the blood. Explain. So therefore, we're going to be getting a so in there somewhere. Explain how the blockage can lead to the death of muscle cells in the heart. Okay. Right, so let's start it off with a bit of description. So there is less or no blood flow to the heart. Okay, so that's what a heart attack actually is. Then, so, let's do blue. So there is less oxygen to the heart muscle. And you've also got so there is less or no respiration. Okay, so what you've done there, and you've done in your explain question, you've then talked about what's actually happening with the heart, and then you've said, so this is then what happens, this is what the problem is. Question number 5, spectacular. Okay, question number 5. This question is about photosynthesis. Complete the equation for photosynthesis, choose the answers from the box. So a plant takes in carbon dioxide, let's turn it green because it's a bit of biology. Plant takes in carbon dioxide and it gives out glucose. What else does a plant need? It needs water. Like glucose, then what else does it give out? It also gives out oxygen. What is the chemical formula for glucose? Well, that's carbon dioxide, that's water, that's oxygen. So C6H12O6. I love it, sounds great. 5.3. Give 2 ways plants use the glucose produced by photosynthesis. Okay, so even though we then take the glucose and sugar and all that sort of stuff, right, 2 ways in which a plant uses it. It will use it for respiration. Okay, so even though you don't think that a plant respires, it does respire. It's respiring all the time because that's what growth and repair is. It will also convert the glucose to starch. So a potato plant, what a potato plant does, it makes the glucose in its leaves. And then what it does then is it transports it around the plant and it goes to the potato and it gets stored as starch. It can be stored as fat or lipids. Okay, like a peanut, right? Like olives, all those different things. And it also can produce proteins from amino acids. Good. 5.4. Right. A person has 2 greenhouses. What a lucky man. In each greenhouse, the plants are all the same type. Great. Receive the same intensity of light and the same volume of water. Sounds good. Grow in the same type of soil. Fabulous. Figure 7 shows the greenhouses. Greenhouse A, greenhouse B. Oh, greenhouse B has got bigger plants. The person burns logs in the wood burner in greenhouse B. There's our log burner, look. Okay. Explain why the plants in greenhouse B grow faster than the plants in greenhouse A. Right, so let's do a describe first. Right, so if you put the log burner in, then what you're going to do is you're going to increase the temperature. I'm going to have to write this a bit small and shorthand. And CO2 due to the log burner. Okay, so it's warmer and there's more carbon dioxide. So there is more photosynthesis. So more glucose is made. So I'm going to go over here. So more proteins are made. Okay, so there's lots of things that you can do there, right? But what you've got to do is you've got to start off with your explain, do a describe first. Question numero 6, right? Drinks contain different substances. A drinks company claims that a drink contains sugar, protein, and fat. Yum, yum. Glucose and fructose are different sugars. Fructose is much sweeter taste than glucose. Suggest 2 reasons why the drinks company uses fructose in the drink rather than glucose. Do not refer to sweetness. Okay, so that's telling you one of the answers you don't write. Right, so less fructose is needed. Okay, so to get the same level of sweetness, right, you need less fructose in there. It is cheaper to produce and buy. That's assuming that fructose and glucose kind of are about the same sort of price, which I don't know if they are or not. Number 3, less calories in the drink. Okay, because what you're doing is you're getting less glucose. Okay, and therefore, so people will buy it because there's less calories. Okay, so describe how a student could test the drink for sugar. Okay, so we've got a sugar test. 3 marks. Give the colour of a positive result. Right, so it is use the Benedict's test. Okay, colour of a positive result. What I'm going to do is I'm going to do it a bit weird. I'm going to go red, orange, yellow, green, blue. Right, Richard of York gave battle, indigo, violet in vain. Colours of the rainbow backwards. Okay, now when it says positive result, what you've also got to do is you've got to remember positive doesn't mean blue because that's what it starts with. So they are the only positive results. The exam question itself, it says 3 marks. So therefore, there must be something else in there. So we've got the test, sugar, we've got the colour. You must heat it to 80 degrees C. That is then your 3rd mark. Okay, 6.3. Describe how a student could test a drink for protein. Protein. Okay, give the colour of a positive test. So this is only 2 marks now. Right, so use the Biuret test. Okay, that's the test for proteins. All right, and the colour of a positive result is purple or lilac. I think the colour lilac. Right, I think the colour of it is great. Oh, 6.4, big question time. Okay, so the human digestive system breaks down proteins and fat in the drink. So we're talking about protein and fat. Okay, describe how protein and fat are digested. Okay, so they're already telling us how we're going to break up our question. All right, you should use the enzymes and where. And the enzymes and where. Okay, so I'm now thinking, right, proteins, enzymes, right, that are then going to be involved. Okay, I've got that. So I know that the protein enzymes, you've got pepsin in the stomach, and you've got protease from the small intestine and the pancreas. This is my little plan in the top right-hand corner. For fats, we've got pancreas and small intestine produce lipase, but we've also got bile, which is where the extra marks come into it. And what bile does, it emulsifies fats to increase the surface area. Okay, so what I've done there is in my right hand corner, and what I normally do is I normally kind of do it in the top right hand corner of my page. Right, so we split it up now and we've got all our answers that we need. So proteins. So proteins are broken down by proteases, and they are found in the stomach as pepsin, and they're also made by the pancreas and small intestine, and they are broken down into amino acids. Okay, so that's our protein bit. Fats is a little bit bigger, and I'm running out of space now. So fats are broken, so that's our kind of first section there. Fats, and what we've done is we've answered that bit, Fats are broken down by lipase, and they are found in, well, they're not really found anywhere, are they? Kind of, they're made in the pancreas and the small intestine. Okay, and what they do is the lipids, which is their fats, are broken down by the lipase into fatty acids and glycerol. Bile emulsifies fats to increase the confident surface area. So then the lipase can then work on it. Right, now it looks like a right mess there, okay, but just remember, do yourself a plan, all right, and then look at what they're asking for, because they're asking about proteins, they're asking about fats, they're telling you about, to talk about the enzymes, they're telling you where, and then what you've then got to do is you've just got to add in extra things to get to those 6 marks. That's a good question. Question number 7. A student investigated the concentration of salt in solution Z. I feel osmosis coming on. Student used a method involving osmosis. Okay. The student used tubing made from a partially permeable membrane. Okay, that stuff there is things called visking membrane. It looks like a sausage. Cut 6 pieces of tubing to the same length. Tie one end of each string to each tubing. Put the same volume of solution Z into each of the pieces of the tubing. So same volume into the tubing. Tie the other end, right? And so you've got a sealed tube. Record the mass of each tube. Place each tube into a different concentration of salt solution after 2 hours. That's quite nice and short. Remove each tube from the salt solutions. Record the mass of each tube. Okay, I don't feel any dabbing going on there. Okay, so it is the osmosis potato required practical, isn't it? What was the independent variable for the investigation? Don't mess in class. Dependent is measure, independent is change. So what are we changing? Right, well, we're not changing the mass of the tube, are we? We are changing the concentration of the salt solution. We're definitely not changing the time, and we're not changing the volume of solution Z, because it says in it that was the same. The student dried the outside of each tube with a paper towel before recording the mass. Why is it important to dry the tube? That is then to remove excess, don't you write excess, liquid from the outside. So excess or extra mass from the water is taken away. Okay, it's a bit like you getting out of the bath, right? If you get out of the bath, you've got loads of water on the outside of you. So what you have to do is get rid of that to get the mass. Ooh, table 3 shows the results. Concentration of salt solution at the start, after 2 hours change. Calculate the percentage change in mass. Okay, calculate the value X in table 3. Give your answer to 1 decimal place. There's 1 mark. Okay, so I'm going to put here percentage change in mass equals the change divided by, this is a nightmare small, start times 100. Okay, so I'm now going to move all the way over here, even though that's not very good. You should try and keep your equal signs underneath each other. So the change is 0.96, which is there, divided by the start, which is 15.54, multiplied by 100, equals 6.1776061. Fantastic. 1 decimal place, 6.2%. 3 marks. 7.4. You should plot the percentage change in mass from table 3 for salt concentrations of only 0.2 to 1. Right, so it's telling you which ones. Draw a line of best fit. Right, so what you got to do now is you got to think that it's telling you, you got to plot, and you got to draw a line of best fit. One of the results have been plotted for you. Okay. Right, now what I'm going to do now is I've kind of pre-done it, so I'm going to go 0.2 goes up to about there. I should have put the table on here really. Okay, 0.4 goes to 2.3, which is there. Now what you also got to remember that plus or minus half a small square loses your marks. 0.6 is there, so no, it's not there, it's not there. Rub it out, make it go away, is to there. And then I've got given that one, and then 1 goes to there. Okay, now I should have had the table on here really, right, make it more clear, right, but what you've got to do is you just got to make sure that you're getting it absolutely spot on in the right place. Now draw a line of best fit. It's going to be quite hard on here. Get yourself a ruler. Okay, looks to me like a straight line. All right, now what I'm going to do is I'm going to mess this up completely. So I'm going to go, oh, damn, that was terrible. Okay, in fact, can I even rub it out? I'm going to rub it out. I'm going to have another go. Here we go. Wow, that's me using a very, very straight ruler. Okay, and what you should have is you should have the same number of crosses on either side. Okay, so obviously mine is way too over this side. Determine the concentration of salt in solution Z. So what you do then is you go to where it crosses, which is this point here, okay, and then what you do is you read it off the axes. Now, if I'd have done mine better, the actual answer is 0.65, so it should have been around about there. Let's just do it in a different color. Okay. Right, now that's the end of the exam. Right, I hope it was beneficial to you. Good luck.